Today's topic is cell wall. Structure of cell wall. Cell wall is basically of three walls, three sub walls. Primary wall, secondary wall, tertiary wall. Primary wall, it is the first layer to be deposited. It is present in all plant cells. It is meristematic cells and parenchyma cells have only primary wall. They do not have any other wall. Primary wall is again divided into two parts, matrix and then cellulose microfibril. Matrix has pectin, hemicellulose and glycoprotein. Glycoprotein arranges or orients the microfibrils. The cellular microfibrils are suspended in matrix, that is, they are parallelly arranged in longitudinal axis. This is a cell wall, and these are the microfibrils. You can see that these are parallel to the longitudinal axis. Growth of primary wall is by process called insusceptation. Insuscep- Insusception. Interception. Intersusception. Intersusception. Intersusception is the increase in size of the wall with in which new material is added into the already existing layer. So, when the water molecules enters by osmosis, then the cell matrix increases and the pectin in the wall becomes soft and slimy due to combination with water. The wall will expand and restore its thickness. It will maintain always its thickness, and the new material is simultaneously added while it uh, increases. Secondary wall also has is present between the primary wall and cell membrane. It has two parts. It also has two parts just like primary wall. Primary it but in here in the matrix no glycoprotein is present. Here only pectin and hemicellulose is present. And due to absence of glycoprotein, cellulose microfibrils are deposited in any directions. Growth of secondary wall is due to deposition of new material over the existing layer. Here no actual it is not formed due to actual making. It is just formed by deposition over existing layer, and that's called new layer. It is also called deposition and or accretion. This increases thickness of wall and decreases volume or lumen of the cell. Every time new layer are reported, microfibrils can have different orientation, like this. In S1 it is like this, in S2 it is like this, and in S3 again it is like this. The secondary wall appears luminar, or that is divided because differentiated because of the presence of different organization of microfibrils. The secondary wall can have 3 to 5 layers, not more than 5 layers. Modification and or chemical changes in the secondary wall for specialization. These are of four types. Again, all these four are just nothing but deposition of four types of chemicals, that is lignin, pectin, suberin, and cutin. Lignin is rigid and compact and thus provides mechanical support. However, it is impermeable to water and then cells are dead at maturity. For example, sclerenchyma and vessels and tracheids. Say vessels and tracheids help in conduction because they are hollow, and that is because they are dead and have empty lumen, so providing space for transport. Pectinization is pectin's decomposition, and it is only found in the stems of dicots, and dicots generally of herbaceous dicots like sunflower who have green stems when they are young plants. These are very weak mechanical tissues, form collenchyma in plants. Suberin, it forms outer dead protective layer of big plants. Cutinization, cutin is impermeable, hydrophobic, elastic, and induces transpiration. Cutinization helps in inducing transpiration. Tertiary wall, it is modified innermost layer of secondary wall, usually after S4 or S5, secondary fourth or secondary fifth layer. Xylem means polymers of cellulose sugar. It is only found in tension wood of tall gymnosperms. It is called tension wood because tall gymnosperm, approximately 50 meters, are very very high and have to pull the great height of the tree and The transpiration also very large amount of water. So to manage that way, tertiary wall is created. Not nowhere else it is found except of tall, very tall gymnosperms, middle lamellae, connecting layer between two adjacent plant cells. It is made up of calcium and magnesium pectates. Calcium is, however, more than magnesium. Pectates have limited water, therefore rigid. In fleshy fruits, during climatic repairing, fruit gradually becomes pulpy due to solubilization of middle lamella. The cells of tissue separate, so tissues become soft. Communication of cells. Communication between cells. Plasmodesmata and pits. Plasmodesmata, it is living continuum, means only in living cells. It is, however, mostly found in parenchyma, collenchyma. It is present in the region that is between primary wall and middle lamella. Suppose if this is the plasmodesmata area, and these are the two cells, and this is the middle lamella. Then this one branches of SER. SER is they connect the two cells matrices, and the part of SER that goes in the symplasm, which is cytoplasm of both the cells from here to here. It is called desmotubule. It connects the SER of two cells, and the transport occurring from plasmodesmata is also called symplastic transport. It will use energy. Pits are unthickened regions of secondary wall. Primary walls and middle lamella are present in dead cells. Pits are of two types, angio and advanced. Not to study much more, pits are only function as pit pairs. That is, if both the pits are only functional, if both the sides are plasma, um, primary walls are absent, and also middle lamella where are absent both the sides. See, here is a blind pit because one side no plasma membrane, but the other side is completely packed. Also, surface view is like this: border pit, primitive and gymnosperms, and primitive angiosperms. Pit membrane has usually a swelling called torus. Torus is a flaccid structure which helps in the um, making the pit membrane flexible. It is formed by the decomposition or deposition of polysaccharides. When water, if for example, moves from here to here, right to left, then the torus moves from here to here, causing this space to reduce very much, and thus water um, is partially blocked, and thus making water flow becomes inefficient for the plant. Transport is of two types: symplastic and apoplastic. Symplastic is of protoplasmic and consists living cells, and ATP required here and done through plasmodesmata and cell membrane. Apoplastic cell transport is done by non-protoplasmic and has non-cellular done in non-cellular space and no ATP required. Example: pits and intracellular space. Plasma membrane or plasmolemma, the name given by Plowe. First name given to it was cell membrane by Nageli, however it was wrong. Also known as biomembranes, plasma membrane highly dynamic as it performs several functions capable of growth, repair, elastic, selectively permeable. Transport helps in identification, recognition of cell, and enzymatic folds, has folds and modification.